Hello everyone, I welcome you all for the Excellent NEET Academy Dharwad online classes. So I am glad to introduce myself as a part of Excellent NEET Academy Dharwad. So I am Srikanth KS, working as biology faculty. So today let us have a clarity about the most important concept, one of the most important concept in the NEET, in the biology. Translation. So it is a concept which comes in the chapter molecular basis of inheritance, the chapter which is having a weightage of around 5 to 6 questions in the NEET examination. So now this topic, translation, is all about where a particular molecule, that is a nucleic acid, RNA, is converted into a polypeptide chain. So here the complexity which arises is there is a conversion of a nucleic acid into a polymer of amino acid, which is totally two different biomolecules, and obviously you cannot get any complementarity between these two molecules, wherein in the previous topic what you might have come across, that is the replication as well as the transcription, there is the clarity about the complementarity between the two molecules. So here we are not going to find out any complementarity, hence it becomes a complex process. So now before getting into the concept of the translation, let us have some clarity. So this is very important. Before understanding or getting into the topic, we need to have the clarity about the topic, what is the need of knowing that topic and in what way we need to understand that topic. So now to understand what is needed, let us have a look at the tools which comes in this process. So the first thing is, as we say, the definition says, it is the conversion of the RNA into the protein. So the first and the foremost prerequisite molecule in this process is mRNA, which basically acts as a messenger molecule. Why do we consider it as a messenger molecule? Because mRNA contains the codons which codes for specific amino acids. Now as I said, it becomes the most prerequisite molecule. So because as it says here, it contains codon which codes for amino acids. Now we are talking about the translation, that is a conversion of RNA into the protein. Now what is protein? It is polymers of amino acids. Since it is a polymer of amino acid, we require a molecule which is going to allow a amino acid to come into that particular place and become a polymer. So let us now look into the second important thing which is required. So it is a non-membrane bound organelle called as ribosome. Now ribosome, why we require this? We would have studied in our first year in the cell chapter. So where we are going to talk about the ribosomes over there. So we have studied about the prokaryotic ribosomes, you have studied about the eukaryotic ribosomes. So you might be knowing that prokaryotic ribosomes will be made up of 70 S subunits, whereas the eukaryotic is made up of 80 S subunits, where S stands for the Swedberg unit. So all these are the basics which is very much necessary before getting into this chapter. So you have to look into those basics, only then you will get the clarity about this. So when we talk about the ribosome, it is the site for protein synthesis. So now this is very important, the site for protein synthesis. So now with this you need to have one clarity over here. So it is this ribosome inside which the proteins are going to be synthesized. That means amino acids are going to form the chain, that is it becomes a polypeptide chain with the help of the peptide bonds. So this happens inside the ribosome. So that means that this particular ribosome is going to read the codons which are present in the mRNA. Now this is the clarity which you need to know. So it reads the codons in the mRNA. So as a process we will come to know how it is going to read, in what way it is going to read. First we need to have the clarity. So now this particular ribosome is made up of rRNA and various other proteins. So hence even this particular type of RNA. So you have three types of RNA: mRNA, rRNA and tRNA. So now as of now we have come to know the mRNA is essential for translation and even the rRNA is essential for translation, right? And now the next requisite is the tRNA molecule, the transfer RNA. It is also called as adapter molecule. Why do we call it as an adapter molecule? So this particular word usually you don't come across in biology. So hence implicating this word into this statement becomes difficult for the student. So let us have a generalized meaning of this adapter. So what it does, suppose you are having a laptop, so you have any electronic device, so which you need to plug in for charging it. So now what happens, there is a specific current which will be flowing out into the particular wire which you have plugged in. So there may be some fluctuation in the board where the current is being generated or where the current is being flowing. So to come over that particular fluctuations, you always have an adapter. So what does this adapter do? So it sends the current which is necessary for that instrument to function. So that is what the adapter does. So this meaning you must be knowing by now. Now the same meaning we are going to implicate into this topic. Now what does the adapter do here? Why the tRNA is called as adapter molecule? Because now here in the entire process you have an ribosome, you have an mRNA, right? So now the role of tRNA is it brings the specific amino acids into the ribosome, so that it could read, this tRNA with the amino acid could read the codon and leave that particular amino acid inside the ribosome. So this is what it acts as an adapter. So specific amino acid will enter into the ribosome. So it brings the specific amino acid into the ribosome so that the process can continue. So now the most important tools we have come across, that is this mRNA, ribosomes and tRNA. Now all together how it is going to function? What are the steps involved in the translation? Let us have a look at it. So you have 4 steps in which the translation takes place. The first one is activation of amino acid. So it's also called as amino acylation. It is also called as charging of tRNA. So I'll give you the clarity why these different names, but having the same sort of meanings. Next you have initiation, followed by elongation, and the process is completed by termination. Now let us look into the first step, that is activation of amino acid, amino acylation or charging of tRNA. What is this particular step? So it is the step in which a specific amino acid binds to tRNA to form a product, that is amino acyl tRNA. So this reaction is catalyzed by the enzyme amino acyl tRNA synthase. Since there is a product which is called as amino acyl tRNA, this process is called as amino acylation. Now since the tRNA molecule receives the amino acid and gets charged up so that it can enter the ribosome along with the amino acid, so this step is called as charging of tRNA. Now the word, that is activation of amino acid. Why it is called as activation of amino acid? So this clarity is very much needed for the entire process to understand, because people tend to mix this particular thing. So these particular statements are given in a different places in the textbook, but mixing up is very important, right? In what way you are going to mix it, that is very important. Now I am going to give you the clarity why this step is called as activation of amino acid, because now here there is a step which is going to take place in this process, that is initiation as well as the elongation. So this is the step where the polypeptide chain of amino acids are continuously being synthesized. Now for this polypeptide chain to be synthesized, what is required is a bond called as peptide bond. Now this is an high energy consuming bond. Now there is a chemistry involved in this concept which we need to know. Energy consuming bond, that means it requires energy. And another clarity is there is no available energy for that bond to be formed inside the ribosome, but it has to be formed inside the ribosome. So now from where this energy is going to come? The amino acids are going to get that particular energy. Hence, this step is called as activation of amino acid. What happens here is a specific amino acids, when it binds with tRNA, it also receives the molecule of ATP, and then it becomes the charged amino acyl tRNA. So hence the name is called as the activation of amino acids. Okay, now let us understand this in a way by considering the structure of tRNA, because here, as I said, the tRNA is one of the prerequisite tool to understand this process. We need to know a little bit about this particular molecule, where exactly this amino acid binds, how this tRNA enters into the ribosome, so all those things, and how this enzyme is synthesized suddenly. So all these things are very much important. Now this is the structure of tRNA, which is called as the clover leaf model, because it looks like the clover leaf in a two-dimensional structure. Whereas when you look at this in a three-dimensional way, it is going to appear as an inverted L form. Okay. Now this particular tRNA molecule is having 4 arms. So let us start from the 5' and end with the 3'. So here at this particular region there is one arm. This arm is called as DHU arm. DHU means dihydroxyuridine, DHU arm, right? Now I would like to give its particular function. Now this is enzyme binding loop. Why do we call it as enzyme binding loop? See, again, for this, the basics of first year is important from the same chapter, that is biomolecules, where you have studied about the enzymes. So you have this particular reaction you have come across where the enzyme binds with substrate and it forms an enzyme substrate complex. So further it forms an enzyme product complex and it releases the product as well as the enzyme. So this is how an enzymatic reaction takes place. Now look at this particular thing. Enzyme and substrate has become a complex here. That means the substrate has to go and bind to the active site of the enzyme. See, I am not dealing anything away from the NCERT. It is there in the first year, which has to be applied in the second year, because molecular biology is totally an application of the biology concept. So now this particular substrate binds to the active site of the enzyme. That means as a substrate, this particular tRNA also should go and bind to the enzyme to catalyze this particular reaction. So now in this particular tRNA, it is this arm or this loop which goes and binds to the active site of the enzyme. This clarity is very much necessary. So you have the next arm, so that is an anticodon arm. So what it is? So this particular arm, it binds complementary to the codon in mRNA. So now we know that mRNA is having a codon which codes for a specific amino acid, and now that amino acid is going to be brought into the ribosome by this tRNA. Now where it is going to lie in the ribosome? So this is the point here. So this particular tRNA, which is having an anticodon arm, it is an anticodon against the codon in the mRNA. Suppose in the mRNA, if the codon is UUU, so this is going to code for the amino acid phenylalanine, right? So for this particular codon in the anticodon loop, there will be an anticodon, that means a complementary codon over here. So there you are going to get this particular thing in the anticodon loop. So now you got a clarity. Suppose if this is the tRNA which is going to bring the phenylalanine, So in that particular case, this codon, this anticodon loop will be having AAA as a codon, and this particular arm, which is called as the amino acid acceptor arm or loop, it's going to have the codon specific for that particular specific amino acid. So when we are talking about the phenylalanine, it will be having UUU in that particular region, so that specifically that phenylalanine is going to bind to that particular region of the tRNA, so that this tRNA gets completely charged and it can enter into the ribosome. Then you have this particular arm, it's called as tRNA, or it's also called as pseudouridine arm, right? So now what is the role of this? The exact role of this tRNA arm is basically not mentioned in the NCERT, but for the clarity I would like to give you that for a ribosome to accept the tRNA inside, this arm plays a very important role. So how? Quite simple. But in a short way I am going to tell you that. So imagine this is the ribosome, and there is this particular tRNA molecule. This is the tRNA molecule. Now this has to enter inside along with an amino acid. So directly it cannot enter. So because there is no particular cell organelle which can easily allow all the molecules to enter, because there will be that particular selectivity about the molecules. So now what happens here is, on this particular ribosome there will be a receptor, and this receptor is called as the tRNA binding receptor. So it is this arm which binds to that receptor, and ribosome recognizes the tRNA and allows the tRNA inside. So this is how it happens. So this is the clarity about these four arms, which is very much necessary for the process. Now quickly I will move on with the initiation. Okay, now let us know about the initiation. So for the initiation, the most important thing is the mRNA. So now this is, imagine that this is the mRNA which runs from 5 prime to 3 prime. So these mRNA is having the codons. So in every mRNA which has been processed, so it's going to have some sequence called as UTR, which is basically the untranslated region, and then the particular codon begins with the AUG. So AUG is the initiator codon, you already come across in the previous topic, right? And it's going to end with some terminator codons such as UAA or UAG or UGA. So this is the termination codon. So after the termination codon, again you will have the untranslated region. So this untranslated region makes sure that the process is going to begin at AUG and the process is going to end at the termination codons. Right? Now let us begin it. So once this particular mRNA is being processed, or it is being produced after the transcription, this particular ribosome, which is going to be in the separate form as a large subunit and as a small subunit. So firstly, the small subunit comes and binds. So let me write few codons over here. So these are some of the codons which are there. So we need to bring the specific amino acids as per the codon. So now the smaller subunit comes and binds. As soon as the smaller subunit binds, the larger subunits also binds and it forms a ribosomal complex. Now this large subunit is going to have three sites E, P and A. According to NCERT, we need to focus on these two sites. This is called as the exit site. Let us focus on these two sites. That is the P site and the A site. So P site is called as the peptidyl site, where the first amino acid is going to come and bind, and A site is called as amino acid binding site. So here the difference is only the first amino acid which is going to come is going to bind to the P site of the ribosome, whereas the rest of the amino acids is going to compulsorily bind to the A site. So now let us look into it. So you have three sites as I mentioned, E, P and A. So here the first amino acid is going to come and bind. So which will be that first amino acid? Obviously the amino acid with the tRNA which is going to have methionine. Methionine. Why? Because AUG as a codon codes for methionine. So in the previous topic you have come across in the genetic codes, AUG has a dual function. It codes, it acts as an initiator codon, it also codes for methionine amino acid. So now here this particular methionine, that is the tRNA which is carrying the methionine, comes and binds. Now what makes this to bind? The knowledge what I told in the just a while ago. It is the anticodon R which is going to have the codon which is complementary to this AUG. That means it's going to have UAC over here, right? So it comes and binds complementary to it, and that particular process ends the initiation. That means the process is being initiated, right? So next what happens, there is this particular A site which is empty now, so it activates the receptors in the ribosome and it brings another particular tRNA. Which is going to carry the amino acid specific to this codon, UUU, so it codes for phenylalanine. Now the most important step takes place over here. So there will be a formation of peptide bond between the amino acid in the P site and amino acid in the A site. So this is catalyzed by the enzyme peptidyl transferase or peptidyl synthase. Now again there is an application question which can be expected here from the biomolecules. So in biomolecules you have studied all enzymes are proteins, all proteins are not enzymes, but there is exception of enzyme which is not a protein, that is ribosomes. So ribosomes are the class of enzymes which are not proteins but they are the ribonucleic acids, that is RNA. Hence RNA acts as a catalyst. So now this peptidyl transferase is basically a ribosome which is not a protein but it is an RNA. So the first peptide bond is formed between the amino acid in the P site and amino acid in the A site. So now this is where the complexity begins. The peptide bond utilizes the energy as I said. So during that utilization of energy, you need to look at it very carefully here. So the amino acid in the P site, the bond will not be broken. The amino acid in the P site moves towards the A site, moves towards the A site. So that creates the empty space in this particular region where in the P site where the tRNA is there. So it has lost that particular amino acid, it has moved towards the A site. This happens because of the peptide bond formation which utilizes the energy. Now at this particular moment you have the tRNA in A site having two amino acids, tRNA in the P site having no amino acids. Now the elongation continues. The elongation continues. How it continues? By the movement of ribosome, which is called as translocation. So movement. Now there is a rule for this movement. Whenever the ribosome moves ahead, it moves 3 nucleotides ahead. Only 3 nucleotides ahead. Whenever it moves 3 nucleotides ahead. So how it looks when it moves 3 nucleotides ahead? Let us have a look at it. So it looks in this particular way. Now you can see what changes have taken place. The tRNA with 2 amino acid which was in A site has shifted to the P site, hence we call it as translocation. The tRNA in the P site without any amino acid has shifted to the E site. So now there is the space in this A site which again activates the receptors in the ribosome, and another tRNA comes with a specific amino acid. So it comes with a specific amino acid. So GGG, it basically codes for glycine. So now again there will be a peptide bond formation between the amino acid in the P site and A site. So again there will be shifting of these amino acids. So in this particular manner, the amino acids are going to be synthesized. So you have the methionine here, and you had the phenylalanine here, and now you have got the glycine here. So in the similar manner, this translocation keeps on continuing, and there will be a chain of polypeptide which is going to be formed, right? That is what elongation is. Now until where the elongation occurs? Until it reaches the stop codon. Until the stop codon arrives at the A site, this elongation continues. So imagine that it has continued, and now let us move on to the last particular concept, that is the translation. Termination of translation. So now let us imagine the ribosome has moved along and it has been able to synthesize this polypeptide chain, and at this particular point you have the ribosome which has moved at this particular position. And you have the tRNA with multiple amino acid chain. So imagine the entire particular codons has been converted into amino acids. So now in this particular A site there is this codon, that is the stop codon. Now the fact is tRNA, there is no tRNA for the stop codons, whereas there are tRNA for all the codons which are there. So we are having 64 codons. Among that 64 codons, 61 are going to code for the amino acids, and the 3 are the stop codons, that is UAA, UAG and UGA, which do not have a specific tRNA. When they do not have the specific tRNA, they do not code for any amino acid. So at this particular moment, there is no amino acid which is going to enter into the ribosome. Now what happens? There is a set of protein factor which comes and binds to this particular region, and this is called as the releasing factor. So it's a set of protein factor. So when this releasing factor comes and binds to the A site, so that initiates the termination. What happens? The ribosomes, both the ribosomes gets separated, the polypeptide chain gets separated, and the mRNA also gets separated from that particular position. Right. So now for the separation of the ribosome and for the binding of ribosome, lastly I would like to give one particular application question from minerals and nutrition chapter in the first year. So where in the minerals and nutrition chapter you have a particular mineral, that is the magnesium, where at the end in that paragraph in the NCERT, the last line of that particular paragraph about the magnesium, it says that it basically performs a structural role in ribosomes. It has been mentioned. What is that clarity? Where is that clarity? Here is the clarity. At the initial stage, what happened? The two ribosomes separately, it came and bound separately. So once the smaller subunit came and bind, then only the larger subunit came and bound over it. So for that binding, magnesium is necessary. Now for the separation, magnesium should move out. For that, ATP is necessary. This is the application question which you can expect over there in the NEET. So the magnesium would have bound over here, Mg2 plus, and this has to move out. For that, ATP has to enter. So hence the entire process of termination takes place where the ribosomes as well as the polypeptide chain and mRNA get separated, and that is the end of translation. This is what we think as per what is mentioned in the molecular basis of inheritance chapter. But in the cell chapter, you have one more fact. So this is what application is all about. So you can expect around 20% of these questions in the NEET, application based questions. What it is, this is not the end of translation. Why? Whatever the polypeptide chain I've got is basically a primary structure of protein. Now imagine that there is insulin which is being synthesized inside our body. The translation is happening inside the beta cells of the pancreas. Just imagine, it is happening inside the beta cells of pancreas, where every day in our body insulin is being synthesized. How? By this particular process. Now insulin is basically a quaternary structure. Now what it is being synthesized is the primary structure. Now what happens? The answer is there in the cell chapter. The function of Golgi bodies. So, which is going to modify the protein structures. Yes, these particular proteins enter into the Golgi bodies to the cis form, comes out of the trans form, and then you have the particular protein which was synthesizing in our body. For example, talking about insulin, only when it goes into the Golgi bodies and comes out of it, it becomes an insulin molecule. So this is how the process of translation works out. See, any topic in biology, you need to have the clarity. So if you have the clarity, even though it is required for us to move a little bit ahead for the knowledge, it has to be done. Because these are the chapters which is totally based on the knowledge and the application skills. So if we gain that particular application skills in us, develop those application skills in us, and then concentrate and understand the concept, only then you can crack the questions in these particular chapter, right? So hope this particular video was helpful. So I would like to conclude this particular session. So we would like to, looking forward to get into touch with you people with some more concepts which are going to be useful for the NEET examination. Thank you.